Hello there, this is Michael Yu, and today I have a very awesome mushroom that you might think I bought this in the supermarket, but it's not. This is a wild Agaricus. Look how it looked like when you collect it in the field. But this is the common mushroom that you can buy in the stores, but in this case, this is a wild one. Right? So you want to know a little bit more about this awesome mushroom? Take a look. And yes, you can eat this one.
Let's talk about this genus Agaricus. Most likely this specimen is Agaricus campestris. Now this mushroom looks like the common white button mushroom that is called Agaricus bisporus. Let's take a look at this genus. Agaricus is a genus of mushroom containing both edibles and also poisonous species, possibly over 200 members worldwide. The genus includes the common button or white button mushroom Agaricus bisporus and the field mushroom Agaricus campestris. This is the dominant cultivated mushrooms on the west, obviously in the world. Both can be found in the field, but campestris is the most common on the field, in the wild. One of the main characteristics is using a microscope, where Agaricus bisporus would have 2 spores per basidia, while campestris would have 4. And actually, this is why Agaricus bisporus has his name. Agaricus bisporus can be white or brown, which in this case we call it portobello mushroom, while campestris is always white. Agaricus campestris has gills that are initially pink, then turn red-brown, and finally dark brown. And the spores print is typically dark brown. The Agaricus bisporus is found in grasslands, fields, meadows, and it is one of the most widely cultivated mushrooms in the world. While campestris, very similar, also found in pastures or grassland after a typical, a couple of days of rain, and also can be found on lawns on suburban areas. One of the most important things is to identify if it's indeed Agaricus campestris and not to get it confused with other similar mushrooms that could potentially be poisonous. Obviously, we want to identify if we can eat these mushrooms, but again, in the field, we could encounter certain mushrooms that look alike, and if we don't have a perfect identification, we could potentially eat these mushrooms that are poisonous. For example, Amanita hygroscopica, or like the pink gill destroying angel. So just imagine this destroyed angel, and then you call as a common name pink gill destroying angel. Obviously, the pink gill is because it actually looked like this Agaricus bisporus or Agaricus campestris. Another mushrooms that we could have is also from the genus Agaricus or Agaricus xanthodermus. This is the yellow stainer. So one of the characteristics from the yellow stainer is that it actually stains some sort of yellow on the top part of the pileus. Again, this is very toxic, and if you sort of like see this mushroom that kind of look like agaricus, or maybe agaricus obviously campestris, then you cut a little bit of the stipe, and then of course you can see if it stains yellow or not. And the other usual suspect that it always get confused is Chlorophyllum molybdentis. Now, with Chlorophyllum molybdentis is kind of tricky because chlorophyllum can grow small or big, and typically we really want to see a mature specimen. When it's completely open and mature, Chlorophyllum molybdentis is easy to identify because the smell and the green spore or green gills. So this is one of the most important things. When you identified a mushroom in the field, you need to see the buttons or like the baby mushrooms, and then when they are completely mature, so that way we can see all the characteristics that we use to identify all these mushrooms. Now, you want to hear something very interesting? All right, there is an Agaricus bisporus strain that had been genetically modified using the technique that is called CRISPR, the Cas9 gene editing technique. The plant pathologist Jinong Yang from Penn State used CRISPRs to inactivate a gene that causes mushroom browning. So it's literally just that. When the mushroom is getting old, it sort of start browning, and obviously you don't want to eat a mushroom that is brown or is getting smushy. Now they use this technique to target the polyphenol oxidase gene family, or also called PPO, which encodes the enzyme responsible for browning. Now you can have this mushroom fresh for a longer period of time, and it is more resilient to a long storage period. It actually get even more interesting. The USDA ruled that the mushroom does not need to be regulated because it does not contain any introduced genetic material. This made it the first CRISPR-edited organism to be exempt from the US government regulation. So in other words, by deleting a handful of base pairs from the mushroom genome, Jank knocked out one of those PPO genes, or in this case the polyphenol oxidase, reducing its enzyme activities by 30%. Yang's mushroom did not trigger USDA oversight because it does not contain foreign DNA from plant pests such as viruses or bacteria. Such organisms were necessary for genetically modifying plants in the 80s or the 90s when the US government developed its framework for regulating GMOs. But this newer gene editing technique that do not involve plant pests are quickly supplanting the old tools. Obviously, in the future, they are going to come out with some regulation, but at this point, we are not introducing any genetic material. We're just modifying um, uh, its own genetic material. And in this case, it was just deleting one gene. So, in other words, by deleting those genes, then it won't produce the specific enzymes that cause browning. Is it that interesting?